today. I've got so much to talk about. There's a lot of interesting things going on. And I think in our notes, we talked about disrupting the disruptor.
Right.
I think that's something that we kind of keyed in on years ago, right? I think you reacted to that. And maybe we could just talk a little bit about that phrase. What does that mean to you? Because I have an idea, and I think that, boy, this is right in the wheelhouse of Profusa. So
yeah, John, you came up with that phrase, and I really, really liked it. And I think, the word disruption and disruptor and revolutionary actually gets talked about a lot and gets bantered around. I think Profusa is in a reasonably unique position to create something that builds off of shoulders of giants. And I think the industry really is, or at least healthcare, is really with a lot of momentum striving toward, more prevention, more monitoring, more user-friendly tools, if you will, on creating a lot of insight. What that disruption, though, really has brought to bear is really setting the foundation, in my mind, that approach is going to be incredibly important and meaningful and valuable. The reason it hasn't actually translated to where people's vision has been about this in the last, I would say, a decade or so, is because it requires the next layer of disruption. And the next layer of disruption on top of that idea is the execution component of being able to measure and do things that actually is relevant to the clinical community. And so I think our approach is that.
Maybe disruption isn't a thing, but a process, a multi-layered process, as you said. That, one of the things that I find that is a problem with a lot of the people that I work with is that they have a great idea, and by the time they push that idea through a traditional clinical development plan, 1 year, 5 years, 10 years, the competition is already breathing down your back. In other words, you're obsolete before you even make it to marketplace.
Right.
It's a little bit like the large language models today. A lot of these people are building on LLMs using, let's say, ChatGPT as the back end, and they come up with these sexy new little companies. But the reality is 6 months from now, the LLM modality itself may be transformed. So, I think that just as a discussion around being an innovator, stepping outside of Profusa, you got to look forward, right? You have to be a visionary, but I think you got to look back. You got to look who's behind you, who's breathing down your back. I think this is an important dynamic. Does that make sense to you?
Yeah, I think that's very, very insightful. And it's where I think this whole idea of disrupting the disruptor becomes really important, because the people breathing down your back are the first layer and the first steps of disruption for something that's actually meaningful. And if you're not constantly building off of what they're doing and trying to disrupt what they've disrupted, if you will,
interesting. Kathy Wood at ARK Invest has talked about this with me, that we're no longer in the world of exponential change. And the reason I'm bringing that up is because the reason you're disrupting the disruptor is because disruption itself is happening so quickly.
Right.
It's not like the 50s or the 60s, where, okay, we get a new beta-lactam antibiotic. The rate of change is incredible. In fact, we've kind of coined the phrase of super or hyper exponential change, because it's no longer just a simple mathematical equation. It's actually AI to the power of AI. That's the new mathematical construct, right? And it's just so quick that the wave you catch is no longer a wave of sort of clinical methodology or clinical practice. It's actually the wave of the wave of change, if you will. So anyway, I want to tell you my story about ICE. Okay, can I do that?
All right, let's do it.
Right. So the thought is, what the heck is going on with ICE?
Yeah,
there are 3 phases of the ICE story. Not phase change, not solid to liquid, but even that's amazing, because ICE floats, right? I mean, when something becomes solid, it becomes more dense, but ICE, when it becomes solid, it becomes less dense and it floats. That's like fundamental. That makes evolutionary biologists flip out, because of the transient dipole-dipole interaction of the terminal hydrogen charge densities that actually let liquid water pack. All right, I'm sorry, I get carried away. So how do we get ice originally? We got ice originally from cutting it out of a lake. Those were lakes up in Pennsylvania where we have a little summer place where they would actually go in there and saw blocks of ice, drag it onto a cart, and then take it to people. And then something came along where we could chemically manufacture ice in ice houses. So we could make ice, but more interestingly, we could make not only ice, but we make dry ice. And dry ice was really interesting because it was functionally dry. It was carbon dioxide, and you can put that into your refrigerator. And as it evaporated, it actually had a phase change from solid to gas. No intermediary liquid phase change there. And so it didn't melt, didn't get wet. It was easy to manufacture. And then all of a sudden, the ice box, as my grandmother would call it, made by frigid air, became obsolete because of refrigeration itself. So we went from guys cutting ice out of the lake, big industry, very, very profitable, to guys making ice and then delivering ice through dry ice principally. And then we went to refrigeration. Each one of them made the prior modality obsolete. But here's the key point, and I know it's a long way to get to this story, but none of them saw each other coming. Nope, the ice fishermen never saw chemical refrigeration coming. And so, that's what happened. Innovation was a function of obsolescence. So I think that we are kind of there in clinical medicine. We talk about, I don't know, I was trying to think what kind of made obsolescence happen. I think of arterial blood gases. That we now have pulse oximetry and how powerful that was. Almost overnight, it made sticking someone in the radial artery obsolete. But where are we now? I mean, this is what I want to discuss with you today a little bit about what is ripe for discussion. What is ice in the clinical world? Is it blood? Is it blood levels? Is it oxygen? Let's go down that road a little bit and talk about this.
John, that's
being indulged in that wacky story.
That's a really, really cool setup. I think it goes back a little bit to what you're talking about earlier, right? Which is disrupting the disruptor from the perspective of that exponential change in the rapid, the rate of change that you're talking about is if you are trying to disrupt the status quo, you're going to be too late. If you have to disrupt,
that's the quote of the day, right? That's the quote of the day. Disrupting the status quo, and a lot of innovators do that,
right? So what you need to do actually is, especially in healthcare, because the innovation cycle is so long and the regulatory cycle is so long. To your point earlier, that disrupting the status quo will put you behind the 8 ball. What you need to do is actually think about how do you disrupt the next layer of innovation, next layer of disruption, so that by the time you're actually getting ready to market, you are still an innovator, as opposed to being relegated to have done something better that somebody else is doing, right? And just an incremental change. Now, so for us, I think you're an incredibly eloquent guy and smart guy, and so I'm going to be thinking about this ice story for a while. So if I'm a little off mark because I'm speaking off the cuff, forgive me.
I didn't tell you about this, right?
So no, you didn't, right?
This is live.
There is no pre-interview on this one.
We always have such good conversations,
right?
And when you're done with that thought, we're gonna go into the new implant as a disruption to CGM and how, in fact, it may not be a disruption. It may be, a curious step. I don't know, maybe sideways. So keep going. This is really interesting.
Yeah. So, listen, I if you kind of think back, and I'm not a scientific historian, although I kind of like to dabble in that a bit. But if you actually think back on why and how has blood become so important, it goes back to the notion hundreds of years ago, humor, right? Because blood is right. Blood really was recognized obviously as a necessary and critical component of life. It was easy to get to, it was easy to handle, it was easy to access. And so a big chunk of the scientific or at least the medical knowledge came from blood, right? Bloodletting, trepanning, et cetera, et cetera.
Yeah.
And as science and healthcare evolves, you're building off of, you know, in kind of incremental advancement, right? Knowledge base, right? What more can you get out of this liquid that you could get from the body easily? And what more information can you get? What more information can you get? All of a sudden, the body of knowledge is really centered around blood and blood draws and what's happening in blood that actually.
Let me interrupt right there, because I think you're going down a really interesting path. That is a path of innovation for many people, right? But I think it's a precarious path. It's a dangerous path. I'm not going to mention the pharma company, but pharma company X has drug Y.
Mm-hmm.
And what they do, what their clinical development program is to expand indications of that drug. More and more, maybe oncological indications. But that kind of stifles innovation in a way, because all they're doing is new uses for the old product. That's like, how can we use blood again and again and again? So I think that it's important, right? But I don't think it's a real innovator's path. I think it's a execution path almost.
Yeah, you know? and I mean, it's a little bit of this urgency versus importance, right? If you actually want to make rapid advancements, it's easier to say, hey, what is a foundation we already have, which is what do we know about blood? And then what more can we get out of it? Because that innovation goes faster from the perspective that you could actually build off of what somebody else have done, right?
But not only can you build off of what someone else has done, it is the phrase that makes me crazy. It's when I introduce a concept to a physician, or to a clinician, or to a scientist, and I say, I want to do, so I don't think you should be measuring blood oxygen. I think you should be measuring tissue oxygen. They look at me and they say, that's not the way I was taught, right? That's not their imprinting. And boy, is that frustrating. And I think that's probably part of what's happening, you know, with Profusa in terms of getting people to think differently. You know, what's his name? Future Shock, the guy who wrote Future Shock, Alvin Toffler. He said the illiterate of this century will not be those who cannot read or write. It will be those who cannot unlearn and relearn. And I think that if there's a mantra for Profusa, is that we have to unlearn the dynamics of PO2, you know, and look at the cellular level. So anyway, go back. You get me so excited. I keep.
No, but John, I think I think what you're touching on here really is the bottom line, right? That if you want to say, hey, what more can we get out of blood? There are a variety of ways and modalities, and people are trying to innovate around how do you actually get blood easier, and then how do you analyze it with greater precision and greater refinement, and what else can you actually measure? I think when you do go down that path, what you miss, right, is why is the vasculatory system in place in the body? And ultimately, for many clinical indications of what are an indication of somebody's health and disease progression is not necessarily around the circulatory system, but it's actually what the cells actually see, what the cells actually get to experience, right? It's like, you know, judging the health of a town by how often does a train run through the train station, as opposed to really looking at the town and seeing what the town is actually doing.
Exactly.
And if you just look at blood, I think many of those measurements are always going to be proxy measurements and probably won't give you the kind of refinement and the accuracy and the indication that you need. And it's where we are really excited about our approach, right? Which is really measuring directly the interstitial fluid, what the cells actually experience, because you take out a layer of a variable, which is what's happening from the blood vessel interiorly in the capillary out to the tissue. If you measure directly in tissue, then you cut out microvascular disease effects, you cut out, you know, pulse oximetry effects, you cut out these vascular effects, because ultimately many of the disease states is what do the cells actually see. And I think that approach ultimately, it may not make blood draws or blood, the blood-borne science, it may not make that obsolete, but I think it does make this other part of the science that we're trying to actually bring to bear in terms of what the cell sees much more important.
So the analogy you used last time was you can measure the watering system, the sprinkler in your front yard, but is the ground wet?
That's right.
Is the grass getting? So where does that take us now in terms of your immediate clinical activity? I want people to get a sense of what Profusa is doing now, and what's available, because I think that it's kind of like using the old commercial, try it, you like it.
Yeah.
Once you recognize the clinical data and the opportunity of looking at tissue oxygenation, and we haven't even got to lactate and other things, which really interest me. But once you recognize that, I don't think you can go back. I mean, once you've tried pulse oximetry, you're not going to stick a needle in that radial artery anymore. I mean, it's almost barbaric. So let's talk about that a little bit, kind of where you are.
Yeah. So our first product that we're going to start shipping in Q1 of next year, we're building on the manufacturing capability, et cetera. And we've announced distribution agreements and end users who are actually going to use it, et cetera, to the marketplace, is in the field of critical limb ischemia. And I think critical limb ischemia is a good example, peripheral arterial disease. And allow me to explain what that means. And it's a good example of, I think, what we're talking about here. So if you have an artery that's blocked and it prevents blood flow downstream, if that happens in your heart, you're downstream of that blockage, the heart muscle dies and you have a heart attack.
Right.
There's nothing magical about the artery in the heart that gets blocked, and arteries get blocked everywhere. So when they have plaque buildup in the extremities, below the knee, in the thigh, femoral artery, or below the knee, the three trunks of the arteries come down, then what you start developing is what's called peripheral arterial disease. And it's actually telling in it of itself, right, that the disease is called peripheral arterial disease. But the indication that comes out is not an arterial indication. The indication that comes out is tissue death, right? So if you have blockage below the knee and you have no more or very compromised blood flow down to the foot, you get neuropathy, and then ultimately there's no nutrients or oxygen going to the tissue in the foot. And then what you see are these very typical, gangrenous toes, and then you have amputations where the wounds just won't heal because the tissue is not being.
Tell me a little bit more about the underlying clinical scenario here. Is this principally a disease of the diabetic?
There's very strong comorbidity or correlation between diabetes patient and this disease population, and usually it's driven by lifestyle, right? Diabetic patients actually, many of them, especially the type 2s, the lifestyle is not conducive to something that's healthier. That's one. And then two, when you develop late stage diabetes, you get microvascular disease, hardening of the arteries and where capillaries don't work. So then not only do you not have enough blood flow, but then the efficiency of gas transport and exchange in the arterial bed is less optimal.
Stop right there, because I want to go down that road a little bit, because this is really interesting, because we measure blood glucose in the blood, right? We measure oxygen in the blood, but there's a whole nother underlying physiology here. When you talk about capillary flow, substrate delivery, that's a whole thing that is kind of cast aside because it's not clinically accessible. It's not clinically convenient. So we defer to a surrogate measurement, right?
Absolutely. And so if you are a vascular surgeon, and this applies a lot of places, but let's just stick with peripheral arterial disease for a moment. If you are an interventional cardiologist or vascular surgeon whose job it is to open up the pipe, open up the blood vessel, to restore the flow down to the extremities, while they could have technical success and they're happy with their tech, because these are all great, incredibly well-trained, talented surgeons, they're going to have technical success. But there's no guarantee that that technical success translates to a clinical success. Because to use the automated sprinkler example that I used last time, just because you open up the pipe, now there's water in the pipe and there's water in blood flow, doesn't guarantee that the grass is going to get wet, that brown patch is going to turn green, because the sprinkler head might not be working, the water may not be getting there. And so that disconnect between technical success and clinical success is what makes these surgeons incredibly excited, because everything.
It's frustrating, right? When you have a technical success. let me ask you a question. I don't even know if I'm supposed to be asking you this, but I'm going to ask it to you anyway. Intraoperatively, when you're actually using the Profusa implants and the reader, is that a tool intraoperatively as well as postoperative to evaluate patency and tissue levels?
Correct. Right.
Okay.
So
could you explain that a little bit? Because that's really interesting.
Yeah. So what happens is inside a human body, we're wonderful beings and incredibly intricate. And there's so many marvelous things that happen in the human body. And because this disease is a chronic disease that took a long time to develop, your body actually develops what's called collateralization. So if you have a blockage in one vessel, over time, the body actually tries to collateralize and trying to still grow vasculature to places that are actually not getting appropriate oxygenation. And so the anatomy, if you will, of the vessel becomes variable from patient to patient. I think that's one. And two, because you have three trunks coming down below the knee, you could have a lesion or a blockage 10, 12, 15, 16 centimeters in one trunk.
Got it.
And then not another. And so the symptoms don't actually come up until you have pretty extensive disease, usually in multiple trunks. So what happens with a physician intraoperatively is, listen, obviously it's a surgery, it's invasive. Every time you're actually punching balloons in the vessel, et cetera, and you're shooting the dyes through, it doesn't help the patients. The dyes are toxic to the kidney. The balloon, you could actually risk rupturing the arterial wall. And so
those walls are crispy, right?
Yeah, right.
I mean, the kind of thing like, it's a precarious, there's the cystic medial necrosis, all sorts of wacky things going on in those arteries, right?
And for these surgeons, if you ask them what they'll say, and by the way, I'm not a physician, but this is just through conversations with them, and what they'll say is that we try to actually minimize the time that we have the patient on the table. So if I open up one lesion, and that's enough, and I have technical success, and I know I have confidence that that wound's going to heal, then I'm done. I could leave other
stop right there. So there are two kind of things here that are interesting to me, and I don't know if your labeling allows this to be articulated promotionally, but from my world, so if I'm a surgeon doing an intervention to reestablish flow, your technology helps me intraoperatively, correct?
Yeah. So listen, at this point, I'm going to say it could, right?
Okay.
So it's set up.
That's fair.
So, right?
That's fair.
Right.
But to me as a surgeon, that's like a really effective tool potentially, because I'm looking at not vascular flow, not warmth, right? Is the leg warm? I'm looking at tissue oxygenation, right? So perioperatively or during the operative process, this is an interesting tool. Then we have post-op, right? So fill me in on that.
Yeah, so intraoperatively, I would actually say the surgeons would say not just an interesting tool, but it's a critical tool. Because while they have the patient in the room and they open up,
here's your disruption, right? I mean, it's not only about just knowing what tissue levels are from a physiologic perspective, but this as an operative tool, advancing my skill. I think it's kind of sometimes a little hidden.
Some of our physicians literally used a phrase, our tool is the GPS for them inside the operating room.
Wow.
It allows them to actually say, hey, this is enough, or hey, this is not enough. I got to keep going. Because even if I have technical success in this branch, or even have a technical success to open up this particular blockage area, it's not enough. I know that I have restored flow, but the tissue is not getting the oxygen. So I know clinical outcomes is not going to be what I expect.
Does that happen, Ben?
Yeah.
I mean, so the vessel is patent, but I'm just not getting that darn flow. I'm not getting tissue. I'm not getting revascularization, if that's the word.
You'll get revascularization, but you won't get wound healing.
Reperfusion.
That's right. Because for whatever is happening down there, whether it's blockage, blockages further down, whether it's actually microparticles that are actually blocking the capillaries, somehow gas exchange is not happening where you actually expect it. And
we see this, quite high. We've seen this in the lab with preservation of ischemic myocardium. When you have ischemia and then you introduce renewed blood flow, you get hyperemic flow. Blood flow 3 times, and that's kind of precarious for the underlying pathophysiology of gas exchange.
Right.
So that's really interesting. So I just want to kind of go back on that. So GPS for the surgical procedure. That's really interesting. So now you've got doctors, surgeons kind of wanting this to enhance their skill,
or decision making, right? What they could actually say, right? Instead of, hey, that worked really well because look, to your point, the foot is warmer now, or I could see patches of pink and it's looking really good, and I could see on the radiography that, hey, the flow is actually restored. It now allows them an additional layer of confidence to say, hey, not only is technically I'm successful, but now clinically I have confidence that the outcome is going to be good, because now I know the grass is getting wet. I know that tissue is getting oxygen, in addition to me opening up the blood vessel.
Prior to the opportunity of using Profusa technology, what did they do? I mean, did they just hope for the best? Basically, that's it, right?
You hate to say it. You hate to say it, but that's it. It is, I've got technical success. Now let's monitor you really closely. Let's see in a couple of weeks, in 3 weeks, through duplex ultrasound, through Doppler radar, if the flow is still there. Let's keep an eye on the wound to see if the wound dressing is helping the wound heal, etc. So it's a guessing game.
Yeah. So clearly now what I'm seeing is the intraoperative utility of this technology is very, very powerful. Now, then we've got the post-operative follow-up. I don't know if it's a week, 2 weeks, whatever, where people start to get nervous again.
Right. And so the post-operative part is actually quite exciting. So if you look at this particular patient population, and it could be translated to others, but if you look at this particular patient population, they're usually a little older. Clearly, because they have injury to their foot and their extremities, they're less mobile. And the current paradigm is, hey, I'm going to discharge you as a surgeon, and you're going to go see a wound care specialist now. And the wound care specialist is not a surgeon. It's a physician, though, that looks at the wound and with dressing and with antibiotics and such to try to keep the wound, make sure the wound actually heals. And oh, by the way, if you're a more brittle case, I'd like to see you in 2 weeks. If you're a less brittle case, I'll see you in a month. And come on in 2 weeks, or come on in a month.
That's horrible, Ben. I'm just going to stop you there. That's caveman medicine to me. I mean, that's like, wound care, bless them for what they do, but they're really going to do wound debridement, sort of peripheral clinical evaluation. And then I got to wait a week or two to go back to the surgeon in special cases when I can be monitoring tissue oxygenation in real time. What am I missing here?
That's right. That's exactly the point, right? And listen, I trying to explain, or at least paint the picture of how it's actually being done today with the utmost respect for the physicians that are involved in this.
Absolutely.
These are incredibly highly trained, highly skilled. Every single one of them have their patient's best interest at heart, and they do the best they possibly can. But I think they're limited by the tools that provide the information for them to be able to do this well. So imagine now that you have a sensor that actually is measuring whether the tissue perfusion remains there. Instead of come on in 2 weeks or come on in a month, the surgeon now could actually have the conversation with the patient. 2 weeks time, I know we have an appointment. Don't bother coming in. Everything looks good.
Wow.
You're less mobile. It's a hardship on you to actually come in. Don't come in. We're good.
So, is this still the thing, Ben?
That is still the thing. Yeah, it's looking a little thinner now.
All right.
Now existing. Yep.
Is it fixed on? Does the surgeon fix it post-operatively, or can the patient move it around on a marker?
The patient could actually do it. The care provider could actually do it. And you don't have to put it on 24/7, right? For this particular application, you take a measurement and say, hey, it's still looking good. And you do it for 10 minutes or so on a daily basis, or every other day, it's still looking good. And so the patient can actually come in. Don't come in. Save the system money. Save yourself some heartache. Save yourself some travel. But I think more importantly, if they need to come in, they don't have to wait for the appointment, because tissue death, right? Time is tissue death. So let's say 3 weeks out, 4 weeks out, the physician can understand and know, wow, or 3 to 4 days out, the physician can understand, wow, the perfusion is no longer there. Hey, Bobby, come on in, Mr. Smith, come on in now, because I don't want to wait 2 weeks, right? Because if I wait 2 weeks, it might be an amputation. If I get you in now, we might be able to do something.
Time, what do we say? We say in with MIs, time is muscle.
Time is muscle.
Right?
That's exactly,
it's not time is money. No, it's time is muscle.
Time is muscle.
In this instance.
That's right. And then on top of that, we talked about this dynamic of a surgeon with a wound care specialist. The surgeon's opening up the vessel, the wound care specialist is making sure the wound could heal. But if a wound is not healing, there are two potential causes, right? One is the wound care, the antibiotics and the dressing needs to be changed.
Right.
The other cause potentially is the patency of the vessel is no longer there. So the pipe is plugged again.
Yeah.
Having this measurement of tissue perfusion in real time takes away this additional layer of potential diagnostics that has to be done if the wound is not healing. Because the surgeon will look at it and says, listen, perfusion is there. It's a wound care issue. And then the wound care specialist could actually focus on that. As opposed to, or the perfusion is no longer there. It doesn't matter actually what you do with the wound care side. We have to open up more vessels. So even post-surgically, it creates much greater and refined information for the right therapeutic choice to be made.
So I'm going to wrap it up a little bit here, but I think that what I've learned, what I find really interesting, is that not only are we optimizing the surgical procedure by actually getting to that, Elon Musk says first principles, right? First principle. Well, what's first principle here? What is tissue oxygenation? Doesn't matter about vascular patency if I'm not getting tissue oxygenation. So that's right. That's interesting, right? That's important. Secondly, just a quick question, Ben. So I'm post-op. When do I, as a patient, start taking measurements with this?
I think the idea is that you could start doing it as soon as you get home, within the first day or two.
And generally, it's once a day, 3 times a day. I mean, you can take as many as you want, right? But is there a protocol?
No, and there's not a protocol yet. I think the beauty of this, quite frankly, right, is the physicians and the patients has a lot of control, a lot of latitude on the frequency and when and how. And I think we leave it to the professionals, in the hands of the professionals, to actually be able to describe how many, how frequent, etc., for them to actually look at the information.
Interesting. And the third sort of prong to this is the wound care specialist.
That's right.
I can see a day in the very near future where I, as not a surgeon, but as a clinician, a medical guy, who is going to mandate that I have this device so I could track what's going on. Because as you said, so perfectly, failure to heal is only sometimes a vascular problem, right? There's an inflammatory component. There's infection. These have intrinsically bad flow anyway. So to be able to differentiate that diagnosis is really powerful. So it makes everybody a hero, right? I mean, it makes the surgeon a hero, it makes the wound care specialist a hero, and it makes the patient much more of a hero. But they're also taking a role in their care because they're checking patency and they're checking perfusion, right? So
at the end of the day, in our minds, right, the benefit to the patient obviously drives all of this, right? Whether it's better outcome, better lifestyle, etc.
Next time, because you're going down the road that next time we're going to talk about the money, because time is money. And
I was just going to
this briefly. Yeah, go ahead. Just jump in.
I was just gonna wrap it up by saying exactly that. There is a level here where it's economically driven, and the cool thing about this, right, is it's not just Profusa. Anytime you could actually create data, reliable data, remotely for a physician, you're actually taking the moment of care away from the most expensive resources in the healthcare system, which is an OR. In the building that's called a hospital, and put it in the least expensive real estate in the chain, which is in a patient's home.
Perfect.
So not only do you actually have all of these benefits and advantages for a patient and for a physician, but from an economic perspective, you're actually doing a lot to take the cost out and give patients and physicians better tools and better care. And I think that's where this is going to be important.
And I'm gonna even push on that a little bit more.
Right.
When you look at diabetic foot, at these conditions, they tend to be in a lower socioeconomic group anyway, right? Is that fair?
I think it's reasonably fair. I think it's,
Mhm.
I think it's fair. Yeah.
I mean, everybody gets diabetes. It's not. It doesn't look at your ethnic background. But I think people have poor access to care. We've
Exactly.
correlated it to a lot of things. That this becomes a powerful clinical modality to keep them out of the hospital. You know, to give them the ability to ambulate, to work. You know, to live their lives. So this is really cool. So we've got like 2 more things to talk about next time, right? We're going to talk about the money.
Yep.
It's fascinating, and I also want to dig deeper on this whole idea of sort of cellular oxygenation, substrate delivery, lactate, all that kind of stuff, because I think that we're seeing this is really the proverbial tip of the iceberg. So,
well, thanks for that heads up. I got to
Yeah.
pull out my old biochemistry book and
yeah,
do a
maybe
little
we'll even
polish.
talk about the Krebs cycle. Remember?
I remember the Krebs cycle. Oh my gosh.
On my wall behind me, I have an autograph from Hans Krebs. I actually met
You're
Dr.
kidding.
Krebs in 1981. Yeah.
Oh my gosh. That's awesome.
And everybody, I mean, everybody always remembers like 1,6-fructose diphosphate and all that
Right.
weird stuff. So we're
Right.
gonna dig into the biochemistry, and maybe I'll show everybody my picture, my autograph of Hans Krebs. So Ben,
That's awesome.
it's always a pleasure. Everybody, I'm John Nosta. Thanks for listening. And Profusa is doing things that are clinically important, clinically valuable, and making a big difference in the world. Take care.